In the first video of DNA, we talked about the important vocabulary words, your double helix, all the different enzymes that were mentioned, helicase, primase, ligase, and polymerase, and so forth, and the roles that they do in the entire process of DNA replication. But DNA replication is broken up into a few major segments: transcription and translation. So we're going to go through transcription now. And then in transcription, we have a few vocabulary words that are pretty important as well. Now we talked about polymerase. We also have what is called RNA polymerase. RNA polymerase is that enzyme that binds to DNA and it separates. Remember that at the very beginning, at the replication fork, that's where the beginning section of polymerase is going to separate it, and helicase again will be the enzyme that does that. And then on the back end of that double helix, which is also the back end of RNA polymerase, it will rejoin together. So RNA polymerase is considered to be the most important of all the different enzymes, because this is the one that does the bulk of the work, if you will, in terms of lining up the nitrogenous bases as they're coming in, aligning them, proofreading, making any adjustments that need to be adjusted, and so forth. So definitely RNA polymerase is a really big deal. Introns and exons are basically the sections that are involved or not involved. Both introns and exons are going to be copied, but then the introns are going to be removed while still inside of the nucleus, so that only the exon is involved in the coding of the pattern for that mature mRNA. And again, transcription, just understanding that is the whole entire process where DNA is making RNA. Okay? Later in the process of translation, that RNA is going to be then made into a protein. But right now, let's just concentrate on transcription, which is the process of basically making DNA into RNA. Now, some important things to remember. DNA is stored in the nucleus, but RNA making a protein does not occur in the nucleus. It actually happens outside on these ribosome areas here of the ER. So your DNA can't leave your nucleus. So it makes a copy of one of the sides, that complementary strand. That is what we end up referring to as that RNA. RNA can leave the nucleus. So RNA is going to eventually exit the nucleus out of here, come here to the ribosomes on the ER, and that is where the proteins are going to be made. This is where tRNA is going to attach to it to make those amino acids. And as amino acids go linking up, you make your protein. Now let's talk about some ways that DNA and RNA are alike and how they're different. RNA, like DNA, are going to be long chains of nucleotides. Okay, that's one of the ways that they are the same. But there's three major differences between RNA and DNA. First, the sugar. The sugar in RNA is ribose, where the sugar in DNA is deoxyribose. Remember, the difference is the one less oxygen in there. RNA is going to be a single strand, where DNA is a double strand. And DNA codes using thymine. RNA does not. RNA doesn't code for thymine. RNA replaces thymine with uracil. Where thymine always goes attached to adenine in DNA, well, in RNA, uracil will always go attached to adenine. So instead of AT in RNA, it would be AU. Okay, you already learned A has to pair up with T. They fit like a puzzle. Well, once we get to RNA, that it has left the nucleus, you won't see a T. You'll see a U. And so then A will go with U. Once you learn A goes with T in DNA, you'll learn that C and G always go together. Then in RNA, that will help you, that C and G have to go together. So by default, A will go with U, uracil. Now there are three types of RNA that we need to be familiar with. You have your messenger RNA, your transfer RNA, and your ribosomal RNA. Your messenger RNA, that's the copy. That's the one that's going to leave. In the video that I put up on the website, the link for it, that 2-minute video that you guys see, that little yellow strand that pops out of the polymerase, that is your mRNA. That's going to be your copy, and that's going to basically float out of the nucleus and basically leave through a section in the nuclear membrane and leave to be adjoined with tRNA. tRNA is the one that transfers the amino acid to the ribosome, and it's going to do that by following the code of mRNA. mRNA, remember, is a single strand, so it's looking, and if you notice here on the picture, it'll show you, it's looking for tRNA to come and attach. Obviously, by the rules of U going with A and C with G, it can't just attach anywhere. It needs to find these specific spots that it does so. And when it does, it's going to break off in sections of 3. Remember that sections of 3 are called codons. So the codon and the anticodon basically pair up with each other, and they break off in a section of 3, and there is the process of making your amino acid. Ribosomal RNA makes up the majority of your ribosome. The proteins are going to be assembled, and there's an upcoming slide that will show that better on the picture. So again, the same explanation that I was just going through here. You have inside your nucleus, your DNA makes your one strand of mRNA. Your mRNA leaves the nucleus and transports over to have the tRNA come and attach to it. Once you have that done over by the ribosome, you have your rRNA, your ribosomal RNA. So, the basic part of how transcription works, which I've pretty much gone through all of it, but just to kind of recap again. RNA molecules are produced by just simply taking the DNA strand and splitting it apart. And then you're going to have complementary sides, and polymerase is going to read one of the strands. When it reads one of the strands, it's going to start linking these nitrogenous bases that enter polymerase, and it's going to start sequencing them. It makes those Okazaki fragments. Remember that it does that in certain sections. The other enzymes are going to assist in the process of keeping that DNA sequence split apart. That's the job of helicase. Then ligase is going to basically assist. Ligase is another enzyme that basically assists in the process of aligning them and linking them back together. Once that copy is made, your transcription process is, the big part of it is pretty much done. At that point, polymerase is only going to release that mRNA to leave the nucleus, join to tRNA to then make the amino acid. Remember that this only happens in certain sections. The sections that it happens in, when that copy comes out, those are known as promoters. RNA polymerase binds to the regions in DNA known as promoters. The site as to where it starts is called the site of origin. There's where your replication fork is going to occur. Promoters are these signals in DNA that indicate to the enzyme where to bind so that they can make RNA there. And also very important to remember that only one of the strands, when that double helix is split apart, only one of the strands is going to be copied. So real quick, and let me get my pen out here so you can help me out here. Again, a certain gene of the following sequence, here you have a certain gene of the following sequence of nucleotides. So, from left to right, what's going to be the mRNA here? G would be together with what? C, and with A? And then with C? And with A, T? And then with G? And with T? And then with C? Sorry. My G's don't come out all that great. What's going to go here? And then here? Now, this is mRNA. First you would do your first transcription here. And then what we have to do is this. You're going to have to replace, since it's an mRNA, you're going to have to replace every T with a what? With a U. So instead of it being a T here, this would really be uracil. The codons are broken into sections of 3. So this would be C, U, G, and so forth. Every single one you're going to get in sections of 3. And it continues, okay? And in the end, this right here, when we continue it, is going to be your mRNA. All right, so again, just to kind of recap transcription here, remember here you have your DNA. Your DNA is going to split apart here. Your replication fork, you have your leading strand and your lagging strand. Leading goes from 3 base to 5 base. Lagging goes in the opposite direction. That's going to make that copy of transcription known as your mRNA. So this is your mRNA here. At this point, as you can see, your T automatically becomes a what? A U. So you have your mRNA leaves the nucleus, your tRNA comes and attaches so that you end up with your amino acids. once you end up with your amino acids and these sections of 3 break off in these codons, in the end, you'll end up with your protein. Sorry, let me go back. I know I clicked this. It's about to, whatever that means. All right, let me pause this for a second. Oh, there we go. Sorry. Okay, this is the link. This link is no longer active, but it does take you to this biointeractive. You'll notice it has a link on the bottom. Anyway, you don't really have to go there because I went through the process of going to it, and there's a 2-minute video that I want you to see. I put a copy of the link on the website just to the right of the homework. So I want you to make sure that you take a look at that. It shows in a 2-minute version the whole process of transcription. All right, now, it's important to remember that in this, the editing part is very important, because the editing part ensures that the mistakes are taken care of before RNA leaves the nucleus. Now, the DNA of the eukaryotic genes contains introns and exons. Both are going to be copied, okay? But what will happen there, as I stated before, is that the introns will then be what? Will then be removed, and the exons are going to be the ones that are coded. Okay, so the introns are basically cut out while they're still in the nucleus, and your exons are basically spliced together at that point. When they are spliced together, you have your final form of your mRNA. Okay, that is basically the process of transcription. On the next video, we will go through translation, and we'll also talk a little bit about mutations as well.